Hi everyone, how are you? Welcome to another session of Global IMMUNOTALKs. Um, I am Elina Zuniga from the University of California, San Diego. Um, if you don't know, we have started these Global IMMUNOTALKs with my friend and colleague Carla Rodling in 2020, and we have had an amazing experience with many members of our community joining as organizers or speakers. And so I'm very delighted today to be here to introduce our global immune speaker today, Dr. Daniel Saban. So, Daniel is a Joseph Wadsworth Distinguished Professor of Ophthalmology, Integrative Immunology, and Pathology at Duke University School of Medicine, and a member of the faculty network in the Duke Institute for Brain Sciences. So he did his bachelor degrees at George Washington University. Then he obtained a master and a PhD at University of Florida. And after that, he moved for postdoctoral training at Harvard Medical School. In 2012, Daniel moved to Duke University, where he has been since. His scientific journey began by studying the long standing problem of how antigen presenting cells are regulated in the immune privileged environment. His approach to take advantage of the eye, where immune privilege is most well characterized, sparked his enduring fascination with the eye as a model organ for studying fundamental immunology. Building on this unique expertise, Daniel's laboratory harnesses the eye's exceptional accessibility now to understand macrophage neuron interactions under physiologic and diseased states. Using a powerful combination of in vivo imaging, electrophysiology, and behavioral analysis, his team can observe and manipulate immune cells and neurons in real time. Studying the mouse retina in this way led to the first direct evidence of in vivo microglia heterogeneity at the functional level, which has implications not only for homeostasis but also for neurodegenerative states. More recently, Daniel's team has turned their attention to the peripheral nervous system, leveraging the cornea's unique properties. This remarkable tissue, densely packed with nerves and in constant dialogue with the immune system, serves as an ideal platform for studying neuroimmune interactions. Through these research efforts, his team is uncovering fundamental principles of neuroimmune communication that will have broad implications for understanding various pathological conditions. Daniel's research, as you may imagine, has been published in highly prestigious journals, and he has been honored with many awards. In 2024, for example, Daniel received the prestigious Arvo Kogan Award, recognizing researchers under 45 who have made important contributions to eye research with substantial promise for future impact. So, Daniel, it's an amazing pleasure to have you here and looking forward to your talk that is entitled A Neuroimmune Niche that Underpins Touch Sensation. And as always, is the format of these global immuno talks. We would like to ask you a question before you share your screen, and is how do you prioritize which research question to pursue in a rapidly evolving scientific landscape? Looking forward to learn your thoughts about that.
Well, Alina, thank you for the kind introduction. It's actually an honor for me to be able to talk to the global immunotox community. It's been such a vital part in we 5 years now, I guess, especially
Yeah.
during COVID, which times were really tough. So of course I applaud you and Carla and of course the rest of the organizers. So the pleasure is all mine. With respect to the question, I don't know how much insight that I can give. I would say the most important piece for me is what excites me. And and
Mm.
then the other piece is the taste. Everybody has different tastes, and what excites you is different for each person. So, but once you're able to lock in on that, then the next step for me was to know as much as possible in the field. And then it becomes a little bit easier because you have somewhere to zoom in on. So if you're not zoomed in, it would seem like things are moving so quickly. And indeed, in some of the fields, it is moving really quickly. Microglia, for example, is just at breakneck speed. But there are other areas that are less crowded and move more slowly. So the important piece is to really be familiarized with the field, and then I
Mm.
think you figure out what the gaps are. Um, they become once you understand the system in the field, you figure out what the gaps are, and that's really the my formula, I think, anyways.
Yeah, I
Hope
love
that
it.
helps.
I love it. It makes total sense. I like how you put it, like, right? You need to be excited and then
Yeah.
study, understand the field, and know where are the gaps so you
Yep.
can contribute to that. That's wonderful. You put
Thank
it in a very easy way, and yeah, it's wonderful. Thank you so much, Daniel,
you.
for
My
sharing
pleasure.
that. And now
Yeah.
we are all ears, so you can share your screen.
All right. Make sure I scare the right screen here. And then put this on presenter mode.
Yes,
And
perfect.
you're seeing the right
I see
seeing the right display.
I see the presenter. Yeah, it's perfect.
Okay, great. So I'll get started. Thank you so
Okay.
much.
Thank you.
Again, it's an honor to be here. So yes, speak today about the neuroimmune niche that we found, which underpins touch. And I'm going to focus mostly on the cornea. This is a little tongue-in-cheek picture here of a child or touching water, just to be cheeky about touch. But you know, touch is quite important, in fact, for biological survival. It becomes really clear in this really elegant body of work done by Chesler et al. This was published in 2016 New England Journal of Medicine. I believe most of the authors were at the NIH. And this individual has a genetic polymorphism or variant that leads to a loss of function of the piezo2 mechanotransduction or ion pore. And of course, the identification and description of this was the subject of a recent Nobel Prize to Artem Patapoutian. And there's some idea about what these pores look like structurally. So you can see on the left, this is what it's thought that a closed channel, and then you can see with a mechanotransduction, a touch that changes the conformation of this pore, now enabling polarization and activation. And so you can see that this is not just mere touch necessarily. It involves mechanosensation, proprioception, and interoception. So this is clearly really important for survival and function. So this is my take of, at least in establishing the context for our work, of the research landscape of peripheral neuroimmune research. What you can see on the left here is almost nothing, in fact, is known about the neuroimmunology underpinning touch. And what I mean by touch, I'm talking specifically here about piezo2 mechanotransduction. And you can see even in the context, generally, there's less known about homeostasis. So when I'm talking here about neuroimmunology, I'm focused mostly on macrophages. That's quite different on the disease side. We know a good bit amount about neuroimmune responses, predominantly macrophages, for example, in causing pathologies associated with neuronal disease. And we also know a good bit about this is really a really quick expanding literature about how neurons actually regulate the immune response and even trigger their immune response. So in the context of infection, for example, and there's also quite a nice literature in non-infectious diseases as well. So what you're looking at here is an in vivo two-photon image of a CGRP EGFP mouse. CGRP basically is labeling C fibers or C neurons mostly, then that's what the density of the cornea is. But you can see how densely innervated the tissue is. And by the way, this is only the central part of the cornea. You can see that it's concave, and that's because that's live two-photon intravital imaging. If you see these hair-like structures, those are the nerve terminals that extend into the epithelium. And then you can see the thicker trunks sort of at the bottom. These are the feeder trunks that are actually coursing through the stroma and then make a right turn or upturn to the anterior and allow these nerves to then penetrate through the basement membrane of the epithelium where they then form these leashes. You can see this really gorgeous vortex or whirl, some of us call it, which indicates where the central cornea is, and that's where your axis of vision is. So a lot of the neurotransmission happens at the level of the central cornea. You can imagine how important that is for vision. And just to get a sense of how dense the cornea is, this is a table I pulled from Marford et al. If you look at the bottom, the number of intraepithelial nerve terminals in the central cornea, let's say approximately 600 nerve termini per millimeter squared, or microns, millimeters squared. And so that is about I don't know, close to at least 1,000-fold more than in the skin. And it's several hundred-fold, I believe, or at least 40-fold more than the dental pulp. And I did some calculations, but essentially that's like fitting 50 people on one tile. So it's almost like a mosh pit, if you want to call it that, ready to sense the environment. And you can imagine why it's so important to be able to sense the environment from the cornea side. It acts as an alarm, for example, so that the host can protect themselves. So this is just by way of showing you some of the circuitry that I'm going to be referring to. You'll have the corneal projecting afferents, and that basically links to the trigeminal ganglia here. So when you hear about trigeminal ganglia, you'll understand that these nerves that I'm talking about and axons project from the trigeminal ganglion. So we focused on a lot of the work on macrophages, and the reason is this is an image of a confocal immunofluorescence, both of macrophages, that's CD206, nerves, which is a broad nerve marker, PGP 9.9, and then you can see Schwann cells as well. But you can see there's a really rich network of macrophages. Sorry about the pixelation here, but essentially you can see the number of these or the abundance of these and also their juxtaposition or close association with nerves. So it makes sense if you want to study the nerves in the cornea in terms of physiology, it seemed like the macrophages were a great place to start. And what we found, and this is also uh, basically supported by previous papers as well, doing lineage tracing. This is the MS4A3 CRE developed by Ginu's lab. This was a Liu paper in Cell. And below that is the FLT3 paper. Let me just move this here. There we go. 2000, FLT3. This is initial work. This is our work, but the initial work was in 2001, showing FLT3 is involved in hematopoiesis And then Slava Eppelman in 2015 used FLT3-Cres to show that these are hematopoietic derived. So in both of these cases, you can see the majority, and sorry, this is quite small, but the majority of these macrophages are fate mapping back to the monocyte. So this tells us that these are not prenatal derived in the adult. These are monocyte derived macrophages. And I think that's important to understand function. So regarding function, um, the next thing we did was to look at the macrophages in the context of single cell RNA sequencing to, of course, give us some information about their transcriptomes. One thing we did here, so this is removal of the cornea for dissection and then enzymatic digestion, but one important piece, we added monocytes from the peripheral blood because we knew that these were monocyte-derived macrophages, and we can use trajectory analysis to try to understand how they be recruited to the cornea and their terminal state. And this is what these cells look like. You can then focus in on myeloid cells. And then, as I said, you can do velocity or trajectory analysis. And you can see sort of what makes sense. You have your classical monocytes here that become non-classical, but you also get this directionality, which converges on this terminal population of macrophages, this cluster 1 here. So that is important because we can then look at this cluster 1 and compare it directly to the classical monocytes and get some important gene enrichment analysis or functionality predictions. And what was really cool when we did this is that, for example, here in Go biological pathway, what we found processes is what we found clearly enriched. This is of 15 pathways, approximately 7 of these, or exactly 7 of these, indicate or predict interactions between macrophages and neurons or macrophages and glia. So you can see negative function of oligodendrocyte apoptotic process. We don't have oligodendrocytes in the cornea. We have Schwann cells in the cornea, but this suggests macrophage glial cell interactions. And you can see a number of other ones. This is complement-mediated synapse pruning, negative regulation of glial cell apoptotic process. So you can see a number of these. So this gave us the clue, or at least the suggestion, that macrophages were perhaps interacting with both neurons or axons as well as glia. So what Sajiro Littleton did, and by the way, he's now a postdoc, but he's the graduate student that did all of this work for his thesis. He decided to essentially use these Schwann cell mice, PLP1 mice, immunolabel for TUJ1, which is nerves, and CD206, and look to see where they intersect. The only place where macrophages intersect with both the Schwann cells and nerves is at the level of the basement membrane. So you can see, and this is not random either. He marked these with white. You can see them throughout the cornea. Again, where the intersection between all 3 of these cells, again, the nerves, the Schwann cell, and the macrophage. So what you can see, if I make a schematic of it to make it more obvious, this is where the macrophage lies here. Again, at this intersection between the Schwann cell and the nerve. And this is where the nerve actually sheds its Schwann cell ensheathment and then penetrates through the basement membrane. And exactly where that takes place is where one sees, or at least we saw, the macrophage. So that led us to ask some questions about what's going on here. We have first, our focus was on the macrophage Schwann interactions. And, I think, and this is really pretty, again, 2-photon intravital imaging done in the cornea by Sajiro Littleton. The blue is second harmonic generation. That tells you where it gets lit by the collagen organization of the collagen fibril. So you see where the stroma is. Everything above the blue is in the epithelium. And so you can see the Schwann cells throughout. Now, I think the important piece of this is that when you study peripheral nerves, it's often thought or just disregarded that there are Schwann cells or glial cells around them. So this is important to note that when you're studying macrophage nerve interactions or immune cell interactions, there's often another cell in between the nerve and the immune cell. So what Sujiro decided to do was quite simple. It was to deplete macrophages. He has its two photon system, so he can follow these mice longitudinally. We used CSF1R antibody depletion, and that worked quite well. You'll see there in a second. And then we followed these mice out for 4 weeks. So we're using PLP EGFP to actually look at these mice. This is your baseline, and this is 2 weeks after depletion. This bottom one received the depletion. These top are representatives of mice that received an isotype control. You can see at week 4, this is ex vivo. Again, this is longitudinal. There's no macrophages where the macrophages are dense in the control. But strikingly, we saw no morphological difference between the Schwann cells in the normal macrophage non-depleted state versus the macrophage depleted state. So it suggested, because we didn't see any morphological changes due to depletion, led us to think or believe or conclude that macrophages, at least at the morphological level, were not playing a role in Schwann cell maintenance. So we went on to test the opposite, to see maybe if the Schwann cells might be contributing to macrophage maintenance. What Sujiro did was to download several single cell RNA sequencing data sets. Here you can see is UMAP clustering and the identification of Schwann cells. So I'm marking that here with these yellow arrows and highlighting it here in yellow. And you can see using violin plots, PLP1, which marks both Schwann cell types, and then MBP, which marks the myelinated Schwann cells. And if you look for hematopoietins that play a role in the maintenance of macrophages, it leads you to focus on IL-34. So you can see this is really the Schwann cells have the most enriched expression of IL-34. Just to show you here, these are trabecular meshwork cells that have a good amount of IL-34 positive. However, these are not actually in the cornea. So that led us to think that Schwann cells are expressing IL-34. It's the other thing we did was look at the trigeminal ganglia, and we saw that Schwann cells were indeed there expressing IL-34. They were not expressing CSF1. In contrast, if we looked at our single cell data in cornea, it was the macrophages and neutrophils that were making CSF1, whereas neither of those populations were IL-34. So this led us to ask the question whether Schwann cells through IL-34 production may be maintaining macrophages, and that indeed is the case. So these mice actually came from Miriam Murad, Melanie Gretter, and Burkhard Becker. The three ended up generating these mice. And you can see in this IL-34 knockout, and here's quantified on the right, you can see a significant reduction of the macrophages in the cornea. So this led us to conclude that Schwann cells make IL-34, and that IL-34 is maintaining or key to maintaining macrophages. What we also showed is that at least during the depletion of macrophages, there was no changes in Schwann cell morphology. So it seemed like it was a unidirectional interaction. So then we next moved to a macrophage nerve interaction question. What are these macrophages doing to the nerves or vice versa? So to do this, we landed on these CX3CR1 Cree mice. You can see the CX3CR1, the fractalkine receptor, we know that it's expressed by myeloid cells. You can see that it's showing TD tomato positive macrophages throughout the cornea. But what we didn't, which was surprising to us, is that you can see that it's also marking a number of nerves. So this is just a 3D movie showing you these cells at different angles. You can see the thick trunks. Now Second harmonic's going to come in. Everything on top in the epithelium is above the blue. So you can see it's really marking these nerves. We proved this with ex vivo confocal microscopy, looking at staining with 2G1 and PGP 9.5. But you can see a good number, not all, but a good number of these nerves are actually being traced with CX3CR1 Cre, and you can see a nice association between those macrophages and those CX3CR1 Cre traced nerves. We were perplexed at this, although there were some reports of CX3CR1 Cre labeling neurons in other tissues, although that wasn't really well described. So what we did was to look at the different Cres. This is the back Cre produced by Genstat in the Nature paper in 2003. This is Stefan Jung's CX3CR1 knock-in with floxing FGFP. So in both of these Cre situations, you can find labeled nerves. However, in direct contrast, if you use the Cre-ER, this is the Jung Cre-ER, and this is also the CX3CR1 knock-in, the Littman knock-in. If you take these Cre-ERs in the adult state and you pulse tamoxifen pulse, you can get beautiful labeling of the macrophages, but no nerves. Similarly, if you look at the CX3CR1 EGFP knock-in, you see macrophages, but no nerves. So what that told us is that the CX3CR1 is actually labeling or tracing a developmental lineage of these nerves. So the next thing that we did, excuse me, the next thing we did was to deplete macrophages. And what you can see, this is the same mouse, just longitudinal. One's baseline, and one is 2 days post depletion. You can see massive atrophy of these CX3CR1 Cre traced nerves. You see that both at these long leashes. Remember, those leashes are the epithelial leashes that then turn north or apically towards the um, ocular surface in the epithelium. That's where the nerve termini are. Again, this is the same mouse. This is 2 days following depletion, and I think it's quite obvious how much atrophy we see in these. So it suggested that these CX3CR1 trace nerves were actually macrophage dependent. What was perplexing at the time was that, when we quantified all the nerves with a broad marker, this is 2G1, for example, this quantitation was done by Anna Matinya at University of Houston. We didn't see any striking loss in the depleted nerves, again, with broad labeling. And we also Sajiro did CGRP, GFP, intravital. So these mark most of the nerves, probably over 80%, I believe. And again, we saw no remarkable difference. So that led us to conclude that this was a special population of nerves. We certainly have data suggesting that these were developmentally linked lineage of CX3CR1 Cre trace nerves. And so we were left with the conclusion that CX3CR1 Cre traces a select population, likely a developmentally linked nerve population, again, that was macrophage dependent, because when we depleted macrophages, those leashes, axons, atrophied selectively. So that led us to ask the question, what are these CX3CR1 traced neurons? Is there something significant about this, biologically important about this? So Sajiro turned to geomics, which is a whole transcriptome atlasing spatial RNA-seq platform. And essentially what we did was to use the CX3CR1 Cre TD Flox to go into the trigeminal ganglia and segment the ganglia and segment out TD tomato positive versus TD tomato negative neurons. And you can see here are the counts showing our selection did pretty well. You can actually go, which what we did is back and to the indexed events and confirm which ones were indeed positive versus not. And when you do that, you can then look at the differential gene expression. It's hard to see these genes, but the first thing that really screamed, by the way, you see the M cherry TD tomato. It says M cherry because it can't differentiate the those genes. But you can see quite clearly that piezo2 pops out, whereas what's downregulated are genes such as TRPV1, TRPA1, calc B, um, other transcripts that were indicative of C fibers. So as you may know, C fibers are polymodal. They're thin, unmyelinated. TRPV1, for example, responds to heat. And then you can see some of the peptidergic or peptides that these express, whereas piezo2 is typically expressed by larger diameter nerves, such as A fibers, for example. And there are other markers suggesting this as well. So this led us to think that this CX3CR1 trace population was piezo2 enriched and unlikely to be a C fiber, more like an A type fiber. So of course we did the validation. Here's your TD tomato. You can see these in the ganglia. This is piezo2, and you can find these TD tomato piezo2 double positive. Now I will say not all of the piezo2s have TDT expression, but when you look only at the TDT positives, you see an overwhelming percent of those TDT positive, which again are CX3CR1 Cre traced neurons, are indeed piezo2. So that made us feel confident about our geomics analysis. And then what we did was to mine some public available data sets out there. Probably one of the most complete and well annotated data sets is a harmonized data set across species from Will Renthal's lab. This is work by Shams Beyond. It was published in Science Advances. And it basically enables us to now take our module scores for TD tomato positive or some of the genes, for example, and look to see which populations they match to get a better idea of what these CX3CR1 Cre traced neurons are. And you can see it pops right here in this area throughout. And these are essentially had been annotated mostly, again, as we had hypothesized based on piezo2 as A delta fibers, again, making us quite excited that we are looking at a unique population. We also took our data set and mined a unpublished data set that we got through collaboration through a UO1 consortium funded by the NIH. This is St. Ledger et al. This is work between Anthony St. Ledger and Brian Davis and others at UPitt, which sequenced hand-picked, back-labeled corneal projecting efferents. And similarly, when we look at our DEGs, and I'm not showing you the heat map plots, but it really hits overlaps with these three populations as well, telling us that it looks like we have something that is akin to an A delta fiber. Fortunately, through this consortium, we collaborate with Igor Spiegelman at UCLA in the dental school, who does patch clamping and EFIS. So what he was able to do is actually patch clamp these TD tomato neurons from the TG versus TD tomato negative. So you can see this is a TD tom positive, and here are some TD tomato negatives. So again, this is CX3CR1 traced. And then he was able to do EFIS membrane properties. And it's quite striking that essentially what he saw was lower action potential amplitudes, a different resting membrane potential, altogether telling us that these A deltas or the TD tomato positives were way more excitable, um, suggesting again that these are in fact A deltas. So that left us with electrophysiological data as well, confirming our hypothesis. So what we have continuing to build our model here is a macrophage that's sitting at the stromal epithelial interface with Schwann cells that are providing IL-34 for docking or maintenance of these macrophages. And uniquely, these macrophages seem to be enabling the penetration of axons into the epithelium of piezo-2 A delta fibers. So in this last segment here, I'm going to talk about their in vivo function. So it turns out it's really challenging to immunolabel piezo-2 in mice. In fact, it's also challenging to use to see these in situ with GFP labeled piezo-2 mice as well. So we had to use other means to identify these cells. And what Sujiro came across was a pretty nice literature using FM143. This is a fluorescent or fluorescent sterile dye. It's a vital dye essentially. It becomes highly fluorescent when it enters the cells. And it's been for some while known to be uniquely entering mechanosensing neurons. That was work done by John Wood's lab, and then separately by Corey David's lab originally. And then most recently, Kara Marshall had shown in vivo situations that FM143 was quite specific for piezo-2 nerves and fibers. So that's what we relied on here at first. So this is a rendering or an image from, again, this is FM143 IP labeled. This is about 24 hours after. And you can see, first of all, you can see some of these fine hairs. These are the terminals that are penetrating through the epithelium. Again, the blue is the stroma with second harmonic. And you can see as soon as this turns, the really nice immunolabeling we can find, or sorry, FM143 labeling we can see, including the central whorl. So at least based on the literature, this would suggest that these FM143 positive vital dye stained nerves, at least a large fraction of them are likely to be piezo-2s. And that seemed to be correct since we don't see the full density of neurons being labeled with this dye. It seems to be very select. Morphologically, it seems similar to what we saw with the TD tomato, and I'll show you that more specifically. So what Sujiro did was, because this dye FM143 had a broad spectra, he had to figure out a way to, or use a very tight filtering method so that he can distinguish the two. And he did that first by looking at the baseline at 400 to 550 wavelength. That is the filter in which he used to specifically look for FM143 so that it didn't overlap with the TD tomato. So you're looking here at the baseline on the left and in the middle, showing that there's no FM143 staining or positivity before he did the IP injection. And yet you can see clearly the TD tomato. This is the CX3CR1 Cree mice. So we felt pretty confident about that. And then this is 24 hours later after he gave the FM143. You can now see, and this is I don't know how small this is on your screen, but you can see really nice labeling. Remember that this is an um, under interpretation of the amount of FM143 because we're using this very fine filtration of the signal. So the next thing we had to do was to look if we can see co-localization of the two to suggest whether these are indeed FM143 positive. So we teamed up with Sina Farsu and his student Zane in Department or School of Bioengineering at Duke, who are world-class experts in image segmentation. And what they did was, Zane did in particular, was to use some anchoring points to basically stitch these together and used a 323 sort of MATLAB 3D affine transformation matrix to align and compute. And on the left-hand side, you can actually see the FM143 co-localization with TD tomato, the TD tomato nerves, CX3CR1 um, Cree lineage trace nerve. So that was pretty exciting. The other thing to note is that FM143 only enters the nerve with pores that are open. So essentially, it has to be an active piezo2 channel for it to actually receive the vital dye. So this actually suggests that not only are these piezo2 nerves, but they're functional piezo2 nerves because they're staining with the FM143. So that was a nice piece of data. The next thing Sujiro did was because we saw specific atrophy of the TD tomato, CX3CR1 Cree TD tomato nerves as a consequence of the depletion, what Sujiro did was to look at a longitudinal analysis of the CX3CR1 Cree axons or leashes following depletion. So you can see what he did. This is baseline before depletion. This is 1 week following depletion. By the way, this big blob is actual TD tomato staining, which hits some of the epithelial cells. But you can see the macrophages are largely gone. Now you can see clearly the atrophy of these leashes. That is maintained out to 2 weeks. In fact, it goes even longer. And there's still no macrophages. Now, at around 8 weeks, you get a return, a recruitment of macrophages where they reestablish themselves. And what's interesting is you can now look at these mice with mechanosensitivity over longitudinal space or time, which you see on the bottom on the x-axis. Essentially, this is like a von Frey. It's called the Cochet-Bonnet, but it's a filament that you use for touching and eliciting a blink response. And the filament can change in length, and that's as it gets shorter, you increase the pressure essentially. So you can get an idea of how much pressure is needed to elicit a blink response, which gives you a direct measure of mechanosensitivity. So at week or time zero baseline, in other words, you can see very little pressure is needed to elicit a blink response. And then this is 1 week post depletion. You can see the increased pressure needed to elicit the blink response. That's maintained out to week 2. And then that begins to return back to normal, not fully back to normal, but closer to normal around 8 weeks when these macrophages come back, suggesting that both the depletion results in the atrophy and the reestablishment of the macrophage niche is coincident with the return of towards normal mechanosensation. And then what he did was to look at other genetic perturbations, not just frank depletion. This is IL-34 knockouts. Remember, IL-34 knockouts, remember that IL-34 is important in the maintenance of macrophages in the cornea. Our data suggested that Schwann cells produce IL-34, and this IL-34 maintains these macrophages at those dock sites. In the wild type, you can see normal pressure to elicit a blink response. If you introduce a hemizygous knockout, so you have one normal copy, this is not statistically significant, but you can see it's trending up. But then clearly in the knockouts, where you have no IL-34, it really requires more pressure statistically to elicit a blink response. We saw a similar consequence in CCR2. This is the chemokine receptor that's required to recruit monocytes even in homeostasis. We showed that these are monocyte-derived cells. So in these knockouts, you can see it takes more pressure to elicit a blink response. And then we also used the CSF1R fire. These mice essentially have a loss in microglia, but there are other macrophage compartments that are still okay. We found, I'm not showing you the image, but there were a normal number of macrophages in these mice. They don't have retinal microglia, they don't have brain microglia, but we don't see any difference in the mechanosensation. So this is really a monocyte-derived macrophage population that's seemingly maintaining this niche. And then lastly, we see quite unique selectivity as we saw with the imaging analysis and the histology in the two photon. So this is work again done by Anna Matinia at University of Houston using an automated video capture to quantify mouse grimace, which is one of the most reliable behavioral pain markers. And using a number of these different topical agonists or inducers of specific TRP channels, namely C fibers. So this would be TRPV1. So you do see, sorry, the depleted is in black. You do see a difference in the response to TRPV1 activity in terms of grimacing, but there's no difference to hypertonic solution. There's no difference in isilin, which is a TRPM8, and there's no difference to AITC. It's a chemical that is a TRPA1 activator. So what it suggests to us is that this is quite selective in terms of the population of neurons or axons that are lost to macrophage depletion. It seems to be highly specific to mechanosensation with some defect in one of the modalities, C fiber modalities, but not all. So in conclusion, our data show or suggest that indicate that monocyte-derived macrophages form this tricellular niche that's with nerves and Schwann cells at the epithelial basement membrane. IL-34, which is expressed by Schwann cells, maintains the corneal macrophage survival. CX3CR1 we showed selectively labels obviously macrophages, but what we show here is piezo2 expressing A delta neurons in the cornea. Macrophage perturbation, whether it's depletion or genetic perturbation, led to a select atrophy of CX3CR1 labeled corneal nerves, specifically those A deltas. And there was prominent deficits in the mechanosensation, but much more over other sensory modalities. So there's some unique selectivity in this niche. So just to bring it together, what we show again is that this tripartite or tricellular niche at the stromal epithelial interface, which enables piezo2 A deltas to penetrate through the basement membrane and elaborate or innervate the epithelium, which is probably how this is working. We think that macrophages are enabling the penetration. We don't know specifically. Another possibility, which we haven't fully ruled out, but our data don't really support, is that macrophages are providing some kind of neurotrophic factor that is enabling the axon growth. We don't think that's the case. What seems to be most likely, and we don't have the data, we can just rule out some of these things, is that it might be a mechanical thing in terms of enabling this penetration through the basement membrane, perhaps some sort of modification in the ECM of those sites, which again enable A deltas to penetrate through. We don't see the same thing with C fibers, and that might make sense because C fibers are thinner in diameter, even when we're talking about without myelin sheaths. A deltas, which don't have to be fully myelinated, but they're still thicker in diameter. So maybe there's a size gating situation that's happening here, which macrophages are contributing to. So these are open questions. And then in the last few minutes or so, what's exciting is that in corneal nerves can be imaged in patients, in humans, using intravital microscopy with infrared um, reflectance. So what you're looking at is the basement membrane. It's called Bowman's in humans, and you can see the penetration and the leashes that we've identified in mice that we've shown in mice. We're not the first ones to show penetration or the leashes for that matter. But what's interesting is that you can see morphologically something akin to what we're seeing in mice with these penetration points. And again, you can see these in humans in normal situations. And so perhaps macrophage-laden piezo-2 enriched penetration can be visualized in humans, and that could be an anchor to understanding these in disease settings. There are a number of corneal ocular surface. There's neuropathic pain. There's dry eye disease. There's diabetic neuropathy as well that may be useful in evaluating patients. And then lastly, our work is certainly not the only one to show myeloid cells are involved in managing or underpinning corneal sensory behavior. It's really nice work showing intraepithelial dendritic cells by Holly Chinnery's lab in TRPV1. There's also work by Fuxin Yu, intraepithelial dendritic cells express CNTF involved in corneal sensory innervation. And there's previous work from Paul McMenamin's lab using CX3CR1 showing that there are interactions between macrophages in the stromal trunks of mice, and that when they're injured, those macrophages dislodge in a CX3CR1 dependent manner. Um, and then there's a slew of many other papers in terms of corneal neuroimmunology. I'm just listing here with PMIDs. If you're interested to check out things across from the neuroimmune reflex to um, to growth factors that have been used to therapeutically treat, and so on and so forth. So please do check those out. And then I guess lastly, before I do the credits, we have a lot of nice work now showing how important macrophages are in ocular health and resilience. So I showed you on the left Sajiro's work, which is currently under review. We have another body of work showing resident tissue macrophages in the trabecular meshwork led by Katie Liu with our collaborator Dan Stamer in helping mentor Katie Liu as well. But this is showing that these macrophages in the TM trabecular meshwork are important for governing intraocular pressure. So this could play a role in resilience against glaucoma. And then lastly, we have a body of work looking at the neuroretina and microglia, and we show that not only are they important in just normal vision, but in the context of outer retinal degeneration, these microglia actually playing a supportive role of the tissue through phagocytosis of debris and contribution of trophic factors to support the tissue, somewhat akin to the disease-associated microglia described by Colonna and Ido Amit, but substantially different in some of the genetic profiles and their function and their protective function. So with that, I'd like to thank my fantastic lab. Sajira Littleton is in the middle here, and our Genu, Anna Matinya, Igor Spiegelman, and Victor Perez. And then I just really want to thank the NIH for this UO1 consortium, which has been absolutely outstanding. So that's it. Thank you very much. I think you are muted, Alina.
Yes,
There you go.
thank you so much, Daniel. Thank you. That was terrific. Wow. Dissecting all this step at the molecular level is wonderful.
Thank you
Thank
so
you so
much.
much. And clearly, there is so much that can be learned from the eye on neuroimmunology. So thank you for leading the way on that. Yeah.
Of course.
Uh, okay, so like always, questions for global immuno speakers are via YouTube. And so here you have a QR code that will bring you directly to the YouTube channel where you can, after the live session, in the recorded video, you can write in comments your questions, and Daniel will answer these questions for you. In addition, I would like to remind you that next week our global immuno speaker is Lilin Ye, who is professor at the Institute of Immunology in the Third Military Medical University in China. And so I hope you can join us next week for Lilin's talk. And with that, I would like to thank Daniel again and thank everyone for joining today and wish you a wonderful rest of the week. Okay, take care. Bye.